I think we can start now. Good morning, dear colleagues. Today we are holding the second session of the forum. The first session, as you know, was devoted to neurosciences and artificial intelligence. Today's session is called virology, epidemiology, and immunology. This issue have always been relevant not only for Russia and the Republic of Korea, but also for all countries of the world, which is shown by the situation related to the pandemic, which unfortunately, no borders. Today we are faced with a type of coronavirus infection, the novel SARS-2 coronavirus. Dear colleagues, an appearance of the SARS-2 coronavirus became a real shock for medical community in the world. Not all countries could react quite properly. However, we should notice Korean medicine could do it significantly better than other countries. The absolute figures of morbidity and mortality can be wrong, but when the medical system is effective, the time gap between detection of the infection and death, which corresponds to the time between the beginning of the disease and death, is usually 27-30 days. This parameter indicates whether the medicine in the country is able to contain the spread of the virus infection or not. As we know, Korean medicine was able to contain the spread of coronavirus. Not all countries were as effective. In some of them, the system of virus detection were not effective. Not effective enough. In many countries, like the United States, the system of virus detection were not effective in springtime, but are much better now. Russia didn't know cope badly with the spring wave of infection. But now there are some features of exhaustion. Nevertheless, the virus spreading is under control in the Korea, and it is only partially, if not completely, is under control in Russia as well. In this situation, we have time to think about future. In that situation, I would like to start our program and to invite Dr. Yu Chung-Win to make a presentation, lecture from urgent R&D overcoming the COVID-19 crisis in Korea. Please.
Thank you for nice introduction. Can I start?
Yes, you can.
Hello, everyone. I'm Bang-Min Song, a professor at Yonsei University and Director General of the Biotech Korea, a newly launched vaccine research initiative in Korean government. And I have a privilege to chair this important session of virology, epidemiology, and immunology. We have an opportunity to listen to experts in both countries, experts in vaccinology and immunology, to share our recent progresses on working and controlling infectious disease, including COVID-19. And I have a privilege to introduce the first speaker, Dr. Ryu Chung-min. He is the head of the infectious disease in Korea Research Institute of Bioscience and Biotechnology. He received PhD from Auburn University, United States, and published more than 160 papers, including PNAS, Nature Protocols, Nature Communications, and he is the team leader of COVID-19 task force in CRIP since March this year. And we have an opportunity to listen to his talk on a lesson from urgent R&D to overcome COVID-19 crisis in CRIP. Dr.
Yeah, thank you for a nice introduction. It is great pleasure to give the my information recently get in such as COVID-19 crisis. So, can I see my slide? Yes, thank you. So, my title a little bit changes. I'm trying to explain in lessons from the urgent R&D to combat and COVID-19 in CRIPS. Actually, I'm also really overview in what's going on in Korea. Okay. Let me briefly introduce our institute. Actually, in CRIP, simply CRIP, Korea Research Institute of Bioscience and Biotechnology. CRIP is established in 1985 in Seoul, and then now moved to Daejeon in center of South Korea. We have two branches, which include Ochang and Jeongeop. And in this year, our manpower are 1,126 people. Our budget is 182 I don't know, I cannot say anyway, million US dollars. Actually, our mission is we need to establish the many research infrastructures is related to biotechnology and bioscience, and also we have to figure out the national agenda, which include COVID-19. In this situation, COVID-19 is really complex, and we don't have much and very simple silver bullets, which means it's really simple solutions. So let me think of what's the simplified this situation and what's we need to to overcoming this crisis. So we have the input, which means we have a lot of the manpowers and technologies, and we try to figure out that this problem which is output. So we solve all problems. So from input to output, we need to systems. So system means I will explain later, actually. We also need to understand in circumstance. Circumstance means in environment conditions. So I will explain the one by one here. First of all, I will explain in circumstance. So when you go back to in this January, the first publication related to COVID-19 is published in January 24 in Lancet. In one patient in Wuhan in China entered in hospitals December 1st. Then they check in for 1 month, and the scientists in China figure out that those viruses is totally different from previous one. So that's why we called SARS-CoV-2s. Right now, especially in Korea, we face the third wave. So a lot of patients and a lot of infectious people, the numbers really sharply increase right now. So input is a scientific solution to figure out these situations. So let me simplify this scientific solution here. So the big problem is the infectious disease is the patient and healthy people is mixed together. We need to separate out the two groups. Then with this really scientific methods, we called diagnosis. So normally we use the PCR as a standard method. For healthy people, we need to apply the vaccines to avoid infections. In patients, we treat therapeutic drugs to heal this sickness. The systems in CRIPS, since the first case is reported in January and this year, our institute make the urgent task force. We call the COVID-19 task force in CRIPS. So I serve the team leader of this task force teams. So we did a lot of works, especially first we try to help the diagnosis companies. So they requested antibodies for the rapid kit development. As you see it here, the several companies is like Seagens and Flexens and Biotech Med. Those companies was supported by our the antibodies or some PCR products. We also tried to develop in our own antibody-based diagnosis kit. In vaccine area, our scientists tried to develop the recombinant COVID-19 vaccines, and then we did technology transfers in May in this year. And I think right now is the filing in IND. And drug development, we collaborate with a company which artificial intelligence aids screening for the drug candidates. We also completed animal model evaluation with the selected the drug candidates. Let me introduce a review about the infrastructure too in CRIPS. As I mentioned before, we have two branches, Ochang and Jeongeop. Those two branches established the ABSL3, which means Animal Biosafety Level 3. So in Jeongeop, we we completed the animal model for Syrian hamster. In Ochang, we developed the primate animal model, which is monkeys. So it takes 5 years ago, we already established these facilities. And now, in this year, this is really helpful for developing the vaccine and therapeutic drugs in Korean companies. In this early this year, it was a really hard situation because the virus isolation is a little bit delayed in Korea. Then we rapidly developed the animal system. So normally it takes 6 months. We completed only 2 months. Then we published in the Co-Fiction Journal of Infectious Disease like this. In this, we also found a very interesting phenomenon is COVID-19 infections induce edema of the blood vessels. In output, based on our animal model development, we also support to vaccine and therapeutic developing companies. For example, here, Celltrion, this company attempt to develop the therapeutic antibodies, which is treatment. So for these companies, we applied 9 monkeys, and this antibody is really effective to control COVID-19. And Genexen is another company which developed in the DNA vaccines. They applied in vaccine in 3 times and moved to our institute, and then we challenged this virus, and then this effectiveness is also really good. Both companies is on the clinical trial right now. For 9 months, I served the team leader of the COVID-19 task force in CRIPS. I got a little bit lesson from this urgent R&D. So let me introduce the problem first. In my mind, it's just the future pandemics in Asia situation is really critical, which means you can see here the above picture is airway hub. So in whole Earth, there are 3 hubs in North America and Europe and Asia. In bottom one is WHO reported infection maps, infectious disease maps. So as you can see here, the North America and Europe are really clear, but in Asia, is a lot of disease, infectious disease always occur. So if you remember, it's back to 10 years. So a lot of the infectious disease is began in Asia because there's a lot of population and transportation is really developed. So in this moment, we must consider a center for forecasting infectious disease in Pan-Asia and Pacific areas. And second problem is most of people in Korea, even in other countries, keep asking about scientists, why do we have any effective treatment and vaccine so far? The simple answer is it really takes a long time and paid a lot of monies, like 10 to 15 years and $1 billion generally. Why? The safety and effective issue is really critical. But the biggest problem is the vaccine, for example, vaccine problem developed speed cannot catch it. Disease spread or remaining speeds, which means once we developed, it will take time, and the disease already disappears. I mean, economically, it's really not matched in infectious disease and vaccine development. To avoid this pitfall, many companies try to alternative approaches like drug repurposing. So what's the drug repurposing? We just, skipping with the really long time screening and the toxicity tests. So right now, the many candidate drugs, including remdesivir, is the selected from the drug repurposing process. So in Korea also, the many drug repurposing drugs is tested under clinical trials now. In WHO also has attempted several programs like solidarity trials. So this is sharply reduced the time to test it in clinical trial phase 3s. In vaccine, many companies, even in Korea, has developed different platforms like a virus itself and nucleic acids like viral vector and protein-based vaccines. And in my situation, I got a couple of lessons what's going on in viral drug and vaccine development in Korea. The first two is the overall lessons. I think in Korea, we really need expand infrastructure of ABSL3 and toxicities. And also, in government, need to shorten process on drug filing and permission. Especially in therapeutic drug development, for example, most of the drug is already developed from other countries, not in Korea. So we must consider in Korean companies developed drug must be deposited somewhere, and we need to test it and screen and find a really good effectiveness from Korean companies. We also need to establish systematic artificial intelligence drug programs. In Korea, in this area, really developed well, but we cannot put together all information into drug screening in artificial intelligence. Vaccine development, we also think of filling the gap for any empty vaccine platform like mRNA. We don't have mRNA vaccine platform right now. So we must also consider about this. And also, even though we have the really good candidate, we have difficulties to test it phase 3 clinical trials, which means we do not have much more number of people to test it at this phase 3. And also, we must consider in other aspects. For example, we just focus on virus, but in serious condition, people like in ICU, many patients also killed by acute bacterial co-infections. we also consider this one. And also, we must to think of the social science impact. And the other one is we did a lot of the One Health approaches. One Health means is we also consider to environment in people and other the zoonotic interaction like animals. Data sharing is also important issues we must consider. Yeah, that's it for my presentation. At the end of my presentation, I'm really impressed. Infectious disease and the prevention. The sample paratus means this is a Latin word is always prepared. This is really important lesson actually. The infectious disease and prevention, we must be prepared. Otherwise, we really definitely fail. Thank you for your listening. Thank you.
Thank you.
Thank you.
For your
Thank you,
nice
Doctor. Dear colleagues, so let me to invite for presentation Dr. Olga Karpova. She is a head of Department of Virusology, Biological Faculty at the Lomonosov University, with the presentation Plant Viruses, New Opportunities in Biology. Please.
First of all, many thanks to organizers for possibility to tell you about our study at Virology Department of Lomonosov Moscow State University. Let us discuss the plant viruses as a new opportunities in biotechnology. In the beginning, I'd like to mention that the plant viruses are often perfectly homogeneous in terms of composition and size, and they exhibit a variety of shapes and wide-ranging stability to pH, temperature, chemicals, and protease degradation. They are easy to accumulate and isolate. The production cost of plant viruses samples is extremely low. However, one of the main advantages of plant viruses usage is biosafety. They are not infections for mammals, including humans. In this connection, plant viruses are a promising tool for development of new biotechnology. Plant viruses can be used and are already used as carriers for functionally active molecules, as platform and adjuvant to present foreign epitope in the development of vaccine candidates, as template for microelectronic devices, as viral vector for production target protein in plants, and so on. And now, research for design of new generation vaccines based on plant viruses are underway at Virology Department of MSU. Certainly, at the department we study a wide spectrum of plant viruses. But one of the favorite objects is widely known virus, tobacco mosaic virus, TMV. TMV is a rod-shaped virus with chemical symmetry. Variants consist of code protein and RNA. We demonstrated the very interesting phenomenon of tobacco mosaic virus thermal remodeling. Heating of TMV suspension leads to transformation of variants to spherical particles. These spherical particles, SPS, consist of thermally denaturated molecules of virus code protein and RNA-free. Moreover, the size of spherical particles is controllable and depends on the initial concentration of virus suspension. Some problem, sorry. sorry for some technical problems. Heating of suspension leads to transformation of, the obtaining process is very simple. The first step is propagation of and accumulation of TMV in plants and isolation of virus preparation in short steam heating of the variants at 94, 98 degrees for some minutes. Mandatory step is spherical particles characterization using various methods and approach as dynamic light scattering or electron microscopy, etc. We have shown the following properties of spherical particles. They are very stable against different external factors. Repeated freezing, thawing, reheating, centrifugation, storage at room temperature. Spherical particles is biosafety. They don't contain nucleic acids, and tobacco mosaic virus is an infection agent of plants. And to repeat, plants and mammals have no common pathogen. In separate series of experiments, it was confirmed that spherical particles are not toxic for animals. Spherical particles are biodegradable and have unique adsorption properties. We have shown that the SPS can form complexes with various proteins or even small viral particles by simple adsorption alone. One of the most interesting properties of spherical particles is their ability to enhance the immune response to the antigen adsorbed on their surface. Spherical particles are highly immunogenic. In other words, have immunopotentiation activity and can work as an effective adjuvant. Based on the properties of spherical particles, we have proposed a technology of recombinant vaccine production. TMV are accumulated in affected tobacco plants. The variants were isolated and purified by differential centrifugation. Spherical particles are formed by TMV variants thermal transition at 94, 98 degrees for several minutes. In the same time, target antigen genetic construct is expressed in bacterial cells. Recombinant antigen is purified and obtained. For in vitro assembly of SPS antigen complexes, spherical particles are incubated with recombinant antigen in water or PVC at room temperature for 20 minutes. As a result, we have spherical particles are coated by antigens. As you know, antigens are usually surface glycoproteins of variants or their fragments. We can say that resulting complexes mimic the pathogen variants, but it is absolutely safe. Using this scheme, we have developed candidate vaccines against rubella virus. Reproductive age women, children, and people with immunodeficiency are the focus group of this vaccine. We designed complexes of spherical particles with antigen containing 4 repeats of epitope A, its region of E1 rubella virus surface glycoprotein. E1 is the main target for neutralizing antibodies against rubella. To watch of vaccine candidate formation, we used immunogold electron microscopy, the right part of slide. Architecture of SPS antigen complexes was revealed by primary antibodies specific to rubella virus and secondary antibodies conjugated with colloidal bond. Preclinical studies of these candidate vaccines were successfully completed. The similar approach was used for development of SARS-CoV-2 vaccine candidate. In this case, 3 recombinant antigens should be presented on the surface of spherical particles. All of them consist of neutralizing epitopes from S1 and S2 domain of spike protein. Certainly, the main focus is on the RBD domain. We believe we will get polyvalent vaccines as a result. On the bottom of the slide, you can see our analysis of vaccine particles by immunofluorescence microscopy with different color fluorophores. Our candidate vaccines was added in the World Health Organization list of COVID-19 vaccines candidates on May 22. The mounting evidence that there is mounting evidence that the plant virus may also have an immunopotentiation effect on antigen not bound with their surface. Modern vaccinology focuses on searching for effective, safe, and low-cost adjuvants. The plant viruses could be promising candidates for contributing to the design of novel vaccines as an immunostimulator factor. We have got data on the adjuvant properties of plant virus particles with various shapes. We use rod-like tobacco mosaic virus, filamentous potato virus X, 2 spherical viruses, cauliflower mosaic virus with DNA genome, and bean-mild mosaic virus with RNA genome, and also we use spherical particles. We talked about them before. Both albumin and hen-egg lysosine were selected as the model antigen. In this work, we demonstrated that spherical particles, mosaic virus, tobacco mosaic virus, and cauliflower mosaic virus exhibit adjuvant effect. They significantly increased total IgG titers to model antigen in comparison with antigen alone, while potato virus X and bean-mild mosaic virus did not show any effective stimulation. As you know, virus and protein-based carriers and adjuvants are to be able to activate a self-immune response. That's why we found it necessary to evaluate IgG titers for adjuvants themselves. The analysis of the immune response to the potential adjuvants provide a new kind of data. Potato virus X, cauliflower mosaic virus, and bean-mild mosaic virus induce strong self-immune response. However, spherical particles and tobacco mosaic virus induce low titers of self-IgG. On the whole, our data did not reveal any clear influence of plant viruses' size or shape, as well as the presence of absent nucleic acids on the humoral immune response to the model antigen. Given adjuvant properties and self-immunity to virus particles, in our opinion, the most interesting objects are spherical particles and tobacco mosaic virus. Our results provide new insight on the immunopotentiation properties of plant virus and can indicate the adjuvant based on plant virus can be considered to be promising tools in further vaccine development. Thank you very much for your attention.
Thank you very much. Thank you. Dear colleagues, let me ask to continue our chairperson, Professor Song Ban Lin.
Thank you for your introduction. Let me briefly remove my face masks to make myself a little bit more comfortable. And I would like to use this privileged opportunity to discuss on the virus-like particle as a pandemic vaccine platform. The COVID-19, I guess, is a kind of a tip of an iceberg of many pandemics to come in the near future. The WHO recently published a blueprint list of priority disease, listing about 10 different virus diseases that would be expected to cause a pandemic in the near future. There are various types of vaccines available so far: live attenuated, inactivated, subunit vaccines, and recombinant vaccines, with pros and cons. And I would like to discuss on the VLP vaccine, virus-like particle, which is devoid of infectious viral genome, and the morphology-wise, it is almost identical to the infectious virus, and therefore can elicit robust protective immune response. The key issue of this VLP vaccine is to assemble the key immunological features of viruses as highly repetitive particular structures. And currently, there are at least 3 different VLP vaccine platforms available, and most of the VLPs are produced by eukaryotes, including baculovirus, yeast, or animal viruses. Because of the difficulties of the folding of the monomeric antigens from E. coli, because the E. coli, the bacterial system is favored because of the scalability and speed of production and with high yields. But the distinctive disadvantage of bacterial system is that it does not allow the proper folding of the monomers and subsequent assembly into the higher molecular structure. And therefore, if we develop a system that allow a folding and assembly of the monomers into regular assembly in bacterial system, that would render the bacterial system as a positive and prime platform for the VLP production. And this slide just shows how difficult a monomeric antigen would fold into a multimolecular assembly. And there are various types of kinetic traps that traps the misfolded monomers and misfolded defective assembly of the monomers. What is required is a kinetic control by a type of a chaperone that would allow the one by one piling up the monomer antigens into very nice shaped VLP. So we require a very robust chaperone system for the assembly of viral antigen, and I would like to mention that RNA could serve as a chaperone, and we christen this function as chaperona, a RNA-dependent chaperone. And the key issue is how to harness the RNA-based chaperone to the VLP assembly. The easiest way is to fuse the antigen of interest with an RNA interaction domain and express this fusion protein in E. coli system. And E. coli cytoplasm, there are various types of RNA that would interact with the RID domain and serve as a chaperone for the folding of this fusion protein. Then the cutting, the removal of the RID domain would render the monomer into a autonomous folding into the virus-like particle. So this is the basic scenario of the RNA-guided VLP assembly as an experimental model. Let me show you our example one by one. Let me start with subunit vaccine, followed by VLP, nanoparticle, and subvirion vaccines, and let me start with subunit vaccines. Influenza hemagglutinin is trimer, and as exemplified by 2009 swine influenza case, the swine influenza spread at phenomenal speed, and therefore the speed of vaccine production is well below is not enough to catch the speed of the virus spread. The conventional vaccine platform, egg-based platform, required 6 months of production, whereas the bacterial system, if successful, can be produced within a month. And we use the chaperone technology to express the hemagglutinin into immunological form, relevant form, and immunized the mice with full protection with high hemagglutinin inhibition titer as well as the neutralization titers with sterile immunization, with sterile protection as exemplified by the lung viral titer. Then we address to the VLP vaccine with an example of norovirus. Norovirus is made of the virus particle is made of 180 monomeric antigens. And there are various types of genogroups we need to develop norovirus. Until so far, there is no commercialized VLP vaccine available, although the Japanese, the company is a forefront on this issue. This briefly shows the EM electron microscope picture of the norovirus VLP generated from E. coli system. And the system is so efficient that after this, the high-density cell culture, the VLP could be produced about 1 gram per liter of the bacterial culture. And this provides after immunization, it provides very strong protective ability. Let me go on to nanoparticle vaccine. For instance, we can use self-assembly scaffold, like a ferritin scaffold, as a kind of scaffold for the display of the antigen of interest on its surface. The beauty of ferritin is that it has not only 3-fold axis, but also has a 4-fold axis at N-terminal and C-terminal, and therefore you can display trimeric antigen on the 3-fold axis and quaternary conformation antigen on the 4-fold axis. This shows the brief mirrors the receptor binding domain displayed on the ferritin, and the dynamic light scattering shows a very smooth and homogeneous type of the nanoparticle. And after immunization, it provides a very strong receptor binding inhibition ability. The trimer interface could be used for the display of trimeric antigens, but tetrameric surface could be displayed, could be used for the display of tetramer antigens, as exemplified the Nipah virus G protein, and the nanoparticle is shown as a very homogeneous, good homogeneous types of nanoparticle. Lastly, I want to dwell on the subvariant vaccines and the scaffold for pentameric assembly of viral antigens. The example I show is flaviviruses, including dengue virus, West Nile, Zika virus, and Japanese encephalitis viruses. All these viruses share a common distinctive feature that the pentameric interface among the icosahedral surface, the pentameric interface provides very strong and very important conformational epitope for the protective immune responses. So just focusing on the pentameric surface, we need to develop a pentameric scaffold. And the scaffold was derived from the cholera toxin B, because this CTB, the cholera toxin B, naturally forms a pentamer and would allow the pentameric the presentation of your antigen. Not only as a scaffold as a pentamer, the CTB is a mucosal adjuvant. Therefore, there is a dual purpose of using CTB. One is pentamer scaffold, but as well as the built-in adjuvant property to increase the vaccine properties. This is just one example of the dengue virus ED3 domain that is shown that is after expression and the one single purification step. This monomeric protein is mostly present as a pentamer, and after immunization, it elicits a strong neutralization activity. So briefly summarizing all these facts, the bacterial assembly of the viral antigens into higher molecular structure by using RNA as a chaperone can be applied for various vaccines, including FMDV, one of the devastating diseases in the veterinary system. And polio is near eradication, but so far polio vaccine is produced by the cell culture of infectious poliovirus. And the next phase, the ultimate phase of polio eradication should rely on genome-free recombinant vaccine as shown here. And flavivirus, dengue virus, and Zika, Japanese encephalovirus belongs to the same family and provides the pentameric assembly platform of the assembly of the immunogenic antigen could be applied for various types of viruses in flavivirus. Influenza, hemagglutinin is a major protective antigen, but nowadays influenza neuraminidase feature tetramer is required, is being recognized as good protective antigen, and EMA actually recommends not only HA but also NA to be included as a major criteria for the immune protection. And Nipah F trimer and Nipah G protein tetramer can be served as an example of the emerging virus of global concern. So let me conclude my talk that by bacterial system using a RNA a chaperone for the assembly of the monomer into a higher molecular structure, this technological innovation would provide a fast delivery platform of low-cost vaccines. Not only unmet need vaccines like norovirus and the hand, foot, and mouth disease viruses, but also it will serve as affordable premium vaccine exemplified by the human papilloma viruses, and fast delivery of pandemic influenza vaccines and delivery of low-cost vaccines can be applied to WHO blueprint list of priority disease. Thank you for your attention.
Thank you, Professor Sombalin. Dear colleagues, let me introduce Professor Andrei Kozlov, Professor of the Vavilov Institute of General Genetics from Moscow and Professor of St. Petersburg Polytechnic University, with the presentation expression of evolutionary novel genes and evolutionary young endogenous human viruses in human tumors. Please, Professor.
Слышно меня?
Слышно.
Слышно, да?
Да.
Dear colleagues, I'm going to talk today about the fundamental issues of evolutionary oncology and evolutionary virology. And earlier I formulated the hypothesis of the evolution by tumor functionalization, which says that hereditary tumors at early stages of progression could play evolutionary role by providing extra cell masses for the expression of genes evolving in DNA of germ cells. As a result of expression of evolutionarily novel genes, tumor cells could acquire new functions and differentiate in new directions, which might lead to the origin of new cell types, tissues, and organs. And this hypothesis has several non-trivial predictions, and today I will discuss 4 of them. And number 1 will be the prediction about the activation and expression of evolutionary novel genes in tumors. And we have been able to describe several such genes. And one of them is PBOV1 gene, a novel human gene which originated in human genome de novo. No such gene in primates or elsewhere. And as you can see from this picture, it is not expressed in any of normal tissue of, on the panel of cDNA from like more than 30 normal tissues, but it is expressed in many tumors. This gene was among 2 genes ever described as de novo originated human genes in the world literature. And it was confirmed by other authors. We also discovered the whole, the evolutionary novelty of the whole class of cancer testis antigen genes, which is represented here at the lower left corner. This is an evolutionary age distribution of cancer testis genes, and this is the whole class of evolutionary novel genes, which are expressed in tumors. And this was also confirmed by other authors, and we also have been able to, evolutionary novelty of this whole class of cancer testis genes was confirmed. And we, by in silico methods, we have been able to describe about 100 genes, evolutionary novel, tumor specifically expressed. We call them TCIN genes. So about 100 evolutionary novel genes expressed in tumors have been described using in silico methods in humans. And see how interesting they are expressed. So some tumors, in some tumors, many TCIN genes are expressed. And some TCIN genes are expressed in many tumors. So these genes can be very interesting candidates for tumor vaccinology. We also described a novel class of non-coding RNA genes, and during the last 5 million years of evolution, these non-coding RNA human genes were predominant novel genes in humans. They even they originated more often than cancer testis genes even. So we can probably make a conclusion that TCIN gene represent a novel biological phenomenon of expression of evolutionary novel genes in tumors. Again, we call these genes tumor specifically expressed evolutionary novel. And of course, we remember that human endogenous viruses play a role in the origin of some human genes. They represent considerable proportion of genomic DNA, and they infected, as shown in this slide, they infected human genome at different periods of evolutionary history. So we were interested in studying how this endogenous retrovirus with different evolutionary age in humans, how they are expressed in humans. So we picked up the youngest endogenous virus, which is HML2, with integration time like 800,000 years ago, and here HML5 with integration time 55 million years ago. And we studied these genes using publicly available HERV database, which allows users to access individual reconstructed HERV elements, including their sequence structure and other features. And we also use databases of which include transcriptomes of human normal and tumor tissues. And see what we found. HML2 is better expressed, as we can see from this slide, in in tumor tissue, in small cell carcinoma of lung. And in lymphoma as compared to normal lymphocytes. And like a mirror image of it, HML5 is expressed better in normal cells of lung, in normal mammary gland, and in normal prostate. So we may conclude from the studies that evolutionary younger hairs are expressed predominantly in tumors, and evolutionary older hairs are expressed predominantly in normal tissue. So we went further to study the evolutionary origin of functions of TCN genes. You know that evolutionary novel genes, they do not usually have functions. So the functions are quite later during the evolution of the gene. So we used fish TC genes. We studied them on the model of transgenic zebrafish tumor models. And we were looking for genes which were expressed predominantly in tumors. We found several hundred genes activated and expressed in fish tumors. And then we compared them with genomes of lamprey and myxine, and we found fish TCN genes. And then we were looking for human orthologs of these genes and found human orthologs of these genes to trace the evolutionary trajectory of the origin of functions. And we found that in some of the orthologs of human genes, progressive functions not encountered in fishes originated. What kind of functions? Lung development, mammary gland development, ventricular septum development, placenta development. These functions never existed and will never exist in fishes. And we found them in humans in genes which originally originated in fishes. And we confirmed that some of these genes, for example, lab gene, this gene originally codes for leptin, a central regulator of lipid metabolism. And this gene also regulates brown adipose tissue development in humans, and the brown adipose tissue does not exist in fishes. And it also participates in placenta development, and here we found its expression in placenta. So the conclusion of this part would be that genes determining progressive traits in humans, some of these genes originated in fish and were first expressed in fish tumors. So this result is actually the direct confirmation of hypothesis of evolution by tumor neofunctionalization as it was formulated by us. And finally, I will present you with another prediction which was made, the prediction about the number of oncogenes in human genome and in other mammalian genomes. This prediction was made when we knew only 12 oncogenes. It was in the 80s. And the prediction is that the number of oncogenes should correspond to the number of differentiated cell types in the organism. And oncogenes, differentiation genes, and tumor suppressor genes should evolve concurrently. So what do we have now? Now, as a result of meta-analysis, we described about 400 differentiated cell types in humans. And different databases, different oncogene databases contain several hundred oncogenes. So I would say it was a wonderful prediction. Again, I will repeat that when we knew about 12 oncogenes and when we thought that there are only 20 oncogenes will ever be discovered, we predicted that at least an order of magnitude large amount of oncogenes which have been described during subsequent years in many laboratories, And this number was evolving in parallel with the increase of the number of cell types in multicellular organisms, as represented in this slide. We went further and we studied the coevolution of oncogenes, tumor suppressor genes, and differentiation genes. And we built, again, the curves of evolutionary age distribution of these genes, and we found that, surprisingly, the curves of oncogenes, tumor suppressor genes, and differentiation genes, they coincide, they almost overlap. And if we statistically study this phenomenon, we will find that hierarchical classification of different classes will show that these genes form a cluster with 100% bootstrap value, so with absolute bootstrap value. So, and again, we will conclude here that the parallel evolution of oncogenes, tumor suppressor genes, and differentiation genes is confirmed. And the number of oncogenes corresponds to the number of differentiated cell type in the organism, as was predicted. And I come to the final conclusion. The final conclusion is that the hypothesis of evolution by tumor neofunctionalization has the predictive power, which is number one demand for the new biological or physical or whatever scientific hypothesis. And number two is that the obtained data confirm our hypothesis of the possible evolutionary role of hereditary tumors. Thank you for your attention.
Thank you very much. Let me pass the word to our Korean colleague. Professor Song Wai-Lee, could you continue?
Yeah. I have an opportunity to introduce the next speaker, Dr. Sung-taek Kim. He is currently the head of genetic virus laboratory at Institute Pasteur Korea. And he received a PhD from Iowa State University in the United States and did postdoctoral research at the University of Wisconsin-Madison. And he is currently a member of Pan-Government Working Group for COVID-19 Drug and Vaccine Development and served as a WHO working group on COVID-19 animal models and serology. And he will be speaking on today the identification of antiviral drug candidates against SARS coronavirus 2 from FDA-approved drugs. Dr. Kim, please.
Well, thank you for a kind introduction. And as you can see, I'm going to just talk about the drug development against the COVID-19. All right. All right, the first slide shows the current status of COVID-19. I will just skip this one. And then these are showing the clinical features. And most of the patients, about just 80%, show some mild symptoms. However, the rest of 20% show severe symptoms, including even death. And then there's the etiological agent of this COVID-19 is SARS-CoV-2. And as the name implies, this virus is very similar to the SARS-CoV-1, which was reported in the year 2002 and 3 in the Guangdong province in China. And this one shows the life cycle of actually just in general, just a coronavirus. And all the steps of viral life cycle occurs within the cytoplasm. And then the SARS-CoV-2 belongs to the beta coronavirus, along with the other coronavirus, including SARS-CoV-1 and then MERS coronavirus. And then, as I told you, the SARS-CoV-1 and the SARS-CoV-2 are very similar to each other by approximately 80%. And then the patients show sometimes some mild symptoms or severe symptoms. But in the case of severe cases, we have to distinguish actually two phases. The first phase is the so-called viral phase. In this case, the viral replication dominates. And in this phase, the drugs that suppress viral replication should be effective. But in the case of a second phase, this is so-called inflammatory phase or cytokine storm phase. In this case, the over-excessive immune response causes a problem. So in this case, the drugs that should reduce the immune response should help. So this slide shows some representative drugs depending on each different phases of the COVID-19. And the left one shows antiviral drugs. The first one is the convalescent plasma and then the monoclonal antibody. And this should work as interrupting the interaction between viral spike protein and an ACE2 protein, which obviously interrupts the viral entry. And then the second drug is nafamostat and camostat, which actually inhibits TMPRSS2 protein. And this protein works as priming the viral spike protein, thereby just inducing fusion between viral membrane and the host cell membrane. And without this drug treatment, the virus somehow enters the cell, and then the viral replication occurs. And then one of the most famous drugs in this COVID-19 pandemic is the remdesivir. And then this actually inhibits the viral RNA-dependent RNA polymerases. And in the second, inflammatory phase, mostly just some steroid drugs should be very effective. One of the very famous one is that dexamethasone. And this slide shows some current status of clinical trials, global clinical trial status. And normally, when you just think about just a disease, we actually consider new drug development. But the drug development is actually, well, it takes more than just 10 years, and then it needs a lot of investment. But even in that case, after those efforts, the success rate is quite low, even just maybe less than the 10%. So in this pandemic situation, this new drug development is not really practical. So we are actually thinking about some alternative method, which is drug repurchasing. So we are actually thinking about drugs which are already proven, and then these new drugs are already developed for some other diseases. And then these drugs all has actually gone through all the new drug development procedures, including basic research, preclinical trial, and all the clinical trials. So the advantage of these drugs is that they are pretty safe in the humans, and also all the pharmacokinetic profiles are pretty well just established. So once any antiviral efficacy is proven, either in vitro and then in vivo, then you can actually just skip all the first parts of the drug development procedure, including just preclinical or phase 1 clinical trials, and goes to directly phase 2 or phase 3. And then once any antiviral efficacy is just demonstrate, then you can go to the just some approval, like emergency use authorization. So in our laboratory, we actually did also some drug repositioning study earlier this year. And we were actually pretty well prepared for this coronavirus research because we were working on the MERS and the SARS coronavirus therapeutic development. And so this slide shows some image-based screening strategy. So basically, the upper one is showing the red one actually just a signal from the host cells nucleus, and the green signal is coming from the viral protein. So actually, once the virus increases, the green signal is also proportionally increases. So this image is quantitatively converted to the dose response curve. And then we can just, from this dose response curve analysis, we can see any efficacy of potential drug candidates. So basically, using this scheme, we actually screened 3,000 FDA and IND approved drug library against the SARS-CoV-1. And then out of this, we selected 35 drug candidates. And then later, we also included 13 drugs, which was suggested by the infectious disease specialist. So this one shows our drug repositioning timeline. So we obtained the SARS-CoV-2 from Korean CDC last February 17. And obviously, before this, we were working on the SARS-CoV-1. And then as soon as we received the virus was propagated for the further experiments. And also, we tried to establish our antiviral screening, the assay, and also optimized. And then once we finish it, we actually screened the antiviral activity against the SARS-CoV-2 out of just the 48 drug candidates, and finally identified the 24 drug candidate. So all this process, when we started from the virus obtaining, it took just about 4 weeks, and then a week later we could just upload our manuscript. So from this slide, I want to just talk about just a little bit about just the results, and then this is the DRC analysis from viral cells. This is one of the most frequently used in the virus research. So as you can see there, when the concentration of the drug increases, the green signal is decreases. So this shows the typical dose response curve of our reference drugs, including remdesivir, and then chloroquine, and then the lopinavir. So we also analyzed the antiviral efficacy of other 45 drug candidates. And then here, I want to just talk about just a few of them. The first one is ciclesonide, and you can see the very nice image strips and then DRC. And then this drug was originally developed for asthma treatment, and this is also inhaled steroid. And then approximately when we just submit our the result, the Japanese group also submitted a very similar study. And in their paper, they talked about the NSP15 protein, which is an endoribonuclease of this coronavirus. And this is potential target of the ciclesonide. So based on these two facts, we actually expect that ciclesonide would just play dual roles in the COVID-19 patient. First, by acting just a direct acting antiviral targeting NSP15, and the second as an anti-inflammatory drug. And based on this result, currently the phase 1 clinical trials is ongoing in Korea. And the second is niclosamide, and then again, as you can see, they're very nice image strip and then dose response curve. And this drug is actually an anti-helmintic drug. But very interestingly, over the past several years, many laboratories in the world have investigated its broad spectrum antiviral effect, even including MERS and SARS. And then coincidentally, the mechanism of action was also revealed last year. The niclosamide actually enhances autophagy, which actually reduces the replication of MERS coronavirus, and including SARS-CoV-2. And so far, the story is pretty good, but the problem is that this drug is not really absorbed well in the body. So we need some drug or reformulation before just further development. So anyway, just based on our result, several just phase 1 clinical trials are ongoing in Korea and then the foreign countries. So then we move to a different type of the screening strategy. The prior one is based on the viral cell. This is the African green monkey kidney cell. But then we try to establish the antiviral screening assay with the human lung cell because drug susceptibility could differ depending on the cell types. So somehow this was a little bit tricky, but we were successful in establishing this antiviral screening assay. And again, this slide shows the DRC of our just reference drugs. So in this case, we just focused on the 24 drug candidates we actually identified from the viral cell screening. And obviously, we saw some changes in terms of drug potency. And then many drugs actually became less potent in the human cells, Cal3. And one of the interesting results was from the chloroquine, in the red one. And then IC50 value in the Cal3 was actually increased by about 10-fold. Which means just less potent in the human lung cell. And when we submit our result, a very similar result was also published by the German group. And they also actually did the very similar things. And they used the Cal3 cells and the SARS-CoV-2. And the chloroquine actually was not effective in inhibiting just SARS-CoV-2 infection in human lung cells. But a few of the drugs was actually just more potent in the human lung cells, including just the remdesivir. Here, remdesivir was a 10-fold more potent in human lung cells compared to the viral cells. And also the camostat and the nafamostat, those two drugs was not really just potent in the viral cells, but very potent in human lung cells. And then actually, we actually did this experiment because we actually just reviewed the paper published the last March by the German groups. And in the paper, they described the TMPRSS2 protein as a potential target of antiviral development. And then in this paper, they tested the camostat, and they showed a very good just in vitro antiviral efficacy. So in the right paper, this is a paper came up from the Korea, and this is very small, the case report, and they're showing some efficacy of nafamostat in the severe COVID-19 patients. So then the question is, what is so different between these two cell lines? And the answer is the TMPRSS2 protein. In the human lung cell, CAL3, the TMPRSS2 protein is expressed pretty well. So then the camostat and then the nafamostat should work pretty well. But in the case of viral, the TMPRSS2 protein is not expressed very well. So most of the viral entry actually occurs via endocytosis. So in this case, in order to fusion between the viral membrane and the endosomal membrane, the endosomal acidification is required, which is actually inhibited by the chloroquine and hydroxychloroquine. So this is why just there are so many so substantial differences between these two cell lines in terms of chloroquine and then nafamostat and the camostat. So anyway, I just talked just a little bit about just the nafamostat and camostat. Here I'm just comparing the DRC between Viro and the CAL3. You can see a very potent antibody efficacy in the CAL3 cells. And then the nafamostat was originally developed for a drug for pancreatitis. And then this drug is also being used as an anticoagulant. And the very important thing is that many just COVID-19 patients actually suffering from blood clotting. And then so I think nafamostat should just help those patients by just preventing such a blood clotting. And also just as working as an antiviral, which actually inhibits viral entry. And then based on our results and others, the phase 2 clinical trials are ongoing in Korea and then other foreign countries, even including Russia. And the second one is camostat. You can also see the comparison between the Viro and the CAL3 cell. The IC50 value was actually higher than the nafamostat, but actually this one is also potent compared to the IC50 in the Viro. This drug is also developed for pancreatitis. And then again, this is a TMPRSS2 inhibitor, so it just blocks virus entry. Well, one of the advantages of this drug is that this drug is available, orally available, so which means that this can be used for the patients in the mild symptoms. However, the nafamostat is the IV injection drug, so it cannot be used easily for the patients with the mild symptoms. So camostat is also being tried in phase 2 clinical trials in Korea and the foreign countries. So this is the summary of what I have talked so far, and then I want to thank the people in my institute, and then Malik Paris at the Hong Kong Pasteur for sharing the SARS coronavirus 1, and then Korean governments and the Korean Society for Infectious Disease for their help and support, and then the very kind advice. Well, thank you for attention.
Thank you, Dr. Kim. Very clear and nice presentation. So I will turn my microphone to Dr. Chekonin to Russia.
Thank you very much. Dear colleagues, let me introduce correspondent member of Russian Academy of Sciences, Alexander Lukashov. He is the director of the Martinovsky Institute of Medical Parasitology, Tropical and Vector-Borne Diseases. He is a coordinator of the section Basic Research for Medicine of the Expert Council of the Russian Science Foundation for the Presidential Programs. With the presentation, understanding the coronavirus and the outbreak, please.
Okay, dear colleagues, thank you very much for your invitation, and today I will share some of preliminary results that we have. So my group have been working on emerging viruses, and while coronavirus outbreak was a big surprise for general healthcare services all over the world, for people doing emerging viruses, it was quite an unexpected event. And we did a lot of genetic studies and field studies on other emerging viruses. So now we wanted to apply this methodology on the novel coronaviruses and coronaviruses in general. One of major genetic mechanisms in, I would say, almost in all RNA viruses is recombination. And recombination was also quite well known for coronaviruses. It played emergence of SARS-2 coronavirus, slightly between bat coronavirus and pangolin coronavirus. And in general, among people doing coronavirus studies, there is a concept of a modular spike. So a spike that changes its domains quite easily. The problem, however, was that many events were described, but there was no holistic image because many isolated events do not always create a general picture. Previously, we studied recombination in the coronaviruses. This is a diverse and ubiquitous group of RNA viruses. One of them is poliovirus, but there is over 100 of other viruses. And we have been all taught at our medical or biology studies that in general, all organisms on Earth follow some kind of evolutionary tree and have a distinct evolutionary history. But when we began to study genetic of circulating the coronaviruses, we saw that it doesn't work at all. What is there instead is a cloud of genes that recombine so frequently that it is not possible to trace evolutionary history of distinct genes for more than a few years. And this genetic pool, which is like these clouds on the screen, exists over the whole Earth. So viruses from Brazil can recombine with viruses from Russia. It happens every few years, not over centuries, but over years. For most of the coronaviruses, half of circulating viruses recombine over 5 years. So half-life of non-recombinant viruses is so short. And then we wanted to apply these methods and this understanding to the coronaviruses. So how do you study a cloud? It's not very easy because a lot of images, or a lot of photographs of a cloud do not really tell you how the cloud behaves. The same for recombination. There are a lot of tools to detect isolated events. These methods are based on phylogenetic conflict. So when in one part of genome we have one grouping pattern and another pattern in another part, but they are again like photographs. They do not give a holistic image. So we had to develop or to modify some methodology. I would not go into technical detail here, but we based our studies on correlation of genetic distances into parts, in different parts of the genome, without any statistical corrections, because statistical corrections are only good when you have isolated events. When you have a lot of recombination events, it's like a shuffling of a deck of cards, where all connections are lost and the statistical methods do not work anymore. So we first tried it on model data, and here are examples how these graphs should look in absence or presence of recombination. On the top left panel is absence of recombination. Genetic distances in 2 regions of the genome correlate perfectly well. And on the bottom right are many ancestral recombination events. You see that there are dots deviating from the central line. They represent viruses that exchanged parts of their genomes. Then we went to coronavirus data. Here are 4 genera of coronaviruses. And first, we looked at recombination within ORF1. It's a major open reading frame of coronavirus genome. Indeed, there were recombination events. Isolated events were well known. What we saw is that gamma coronaviruses are combined so often that they effectively create a cloud. You can see it on the bottom left panel, where on short genetic distances, there is hardly any correlation between them in different genome parts. So the viruses recombine every few years. It wasn't like that for other coronaviruses, specifically for alpha and beta coronaviruses. I would remind that SARS-2 coronavirus is beta coronavirus. There are many recombination events, but not exactly a cloud within the non-structural genes. Then when we look at recombination between non-structural and the major structural gene, a spike or S gene, which is the major antigen and basis of most vaccines, we can see that in all coronaviruses there is some sort of cloud. It has different genetic range. The genetic range can be very high in alpha coronaviruses and much shorter in beta coronaviruses. But in any case, these parts of the genome, spike and all other non-structural genes behave on evolutionary scale, on global time scale, and on the time scale of hundreds of years on Earth as almost different entities. Then when we looked within spike, for example, recombination between domains of spike S1 and S2, we saw that there is also quite significant recombination, and again in avian gamma coronaviruses, it was like a cloud. So everything recombining with everything and very often, and much less in alpha and beta coronaviruses, but still quite a significant number of recombination events. When we then looked at recombination within S2, so within a domain of spike, to our surprise, there was much less recombination, almost no recombination in alpha and beta coronaviruses, and significantly less recombination in gamma coronaviruses on a short time scale. So there was a lot of recombination involving distinct domains, but not within domains of spike. Then we, again, I would cut technical detail. We wanted to plot these recombination events over the genome. And the top 4 panels are classical methods for studying recombination. They work in many cases quite well, but you see that for gamma coronaviruses, they failed because recombination was so frequent that it destroyed any statistically significant signal. And it wasn't possible to measure a signal when all the data is so completely shuffled. What our method does, this is the bottom panel, it measures not signal but chaos. And when we measure chaos, it works much better for most, I would say, coronaviruses, but especially for gamma coronaviruses. You can see very clearly, so at the bottom is the genome, the coronavirus genome. You can see how spike is recombinant compared to other parts of the genome. But again, it's hard to see it on a slide, but if you look more closely, it's possible to see that the domains of spike do not behave the same. It behaves as a modular spike. So what we came to is that coronaviruses have distinct recombination profiles in different genera, and it is indeed modular spike, but again, the patterns differ among coronavirus genera. And it's not exactly clouds as we saw in other viruses, but it's more like clouds from a computer game where the clouds are made of blocks like a Lego game. And also what we saw is that the repertoire of receptor binding domains in nature is far greater than of other genes. And it's very likely that the next coronaviruses would emerge by recombination too. Very briefly, another direction of our research. So first we spoke about the virus, now about the outbreak. Why it was so hard to understand and to contain this outbreak. From epidemiology textbook, we know this chart. This is a standard model of epidemic, susceptible, infected, and recovered. First, everybody is susceptible, then the population becomes infected, then recovered, stop circulation of the virus. And all this doesn't work with coronaviruses. Not everybody is equally susceptible. About 30% have T-cell clones active against novel coronavirus. Also, a huge fraction of infections are asymptomatic, so we do not really know who was infected and who was not. And we know that repeated infections are possible, but we do not know at what rate. So now we have to really rework and modify epidemiological models for coronavirus infection. What makes it even more complicated is that real people differ from the molecules in gas from physical models that were then brought into epidemiology. Some people have many contacts, they are socially active, or they do not protect themselves. Some are more isolated. So when we want to understand what level of herd immunity is needed to contain the pandemic, here we mean herd immunity as either induced by vaccine or by a natural course of event. We need to consider the structure of population, and this work is still in progress. The problem is that it's not like on this simple chart, it's more like this. So it's very hard to adapt epidemiological models on this population structure, but we are working. And for future directions of research, of course, now to understand how to end this pandemic, our key interests are clinical trials and understanding of both antivirus immunity and reinfection rate, host factors of disease severity, and so on. At our university, at Sechenov University, we have a clinical trials center, which has now in general 74 ongoing clinical protocols. 11 of them are trials on novel coronaviruses, including trials of vaccine, trials of favipiravir, and the novel IL-6 inhibitors, which were all successfully concluded by now. At the moment, we have around 1,000 beds for COVID patients. The partners of clinical trials center include renowned companies like AstraZeneca, Novartis, R-Pharm, one of the biggest Russian companies. So I think that in this direction, we will be looking forward for future cooperation, but of course, it requires funding because clinical trials are really expensive. Thank you very much for your attention. I would like to thank my colleagues who helped with the work and the Russian Foundation of Basic Research Grant. Thank you.
Thank you very much for a very spectacular presentation. Dear colleagues, we have time for the break probably, and we will be back in 15 minutes. Thank you.
So we come back to our business. I hope everybody is well refreshed by the coffee break. The first speaker of the second session, I would like to introduce Dr. Mi-Hyun Kim, the principal researcher at Korea Research Institute of Chemical Technology. Dr. Kim received her PhD from Korea Advanced Institute of Science Technology, and she published more than 50 research papers on antivirals of infectious disease. She is currently at the antiviral research group of CRIT, Korea Research Institute of Chemical Technology, and she has studied mechanism antiviral compounds against influenza virus, HCV, and now SARS-CoV-2 as well. And she will be speaking on the discovery of antiviral agents against respiratory viruses. Dr. Kim, please.
Yeah, okay. Hello, my name is Mi-Hyun Kim. I'm very happy to have talked this precious joint Korea and Russia joint symposium. Okay, I will start my talk. Today, my presentation is about antiviral activity of nilidrin as an entry block of influenza A virus. Before starting my presentation, I would like to introduce to you my research in my institute, CRIT, briefly. Korea Research Institute has been founded in 1976 with aim to contribute to Korea economic development on the basis of chemical technology. And 2000, we have Korea Chemical Bank in CRIT campus. This year we have a 44th anniversary. This slide shows the organization of CRIT. We have 4 main research divisions. I belong to the Infectious Disease Research Center. So my research aim as a virologist is to find or discover of antiviral agents against infectious diseases, particularly viruses, infectious viruses. So we have very diverse infectious human viruses, so such as influenza and corona and picorna viruses. So my main object is to find antivirals against influenza virus and corona, including SARS-CoV-2, and particularly rhinovirus belong to picornaviridae family. And in my institute, we have biosafety level 2 as well as level 3. This slide shows the standard process to find antiviral agent in my institute. We have, as I said before, we have our own chemical bank composed of about 300,000 small chemicals deposited. Most of part of the compounds have been deposited by domestic organic chemists. At the beginning step, I start the antiviral screening using about 700,000 or 10,000 compounds to select and to screen antiviral hit compounds. After finding the hit compound, I recollect their derivatives from a chemical bank, and then we perform second screening to find their core skeleton to express antiviral efficacy. And at this step, we name this active compound, name this compound hit compounds. At this stage, we evaluate their antiviral activity using another antiviral system, and evaluate or find their target molecule, cellular or viral molecule, and also define their mode of action through collaboration with molecular modeling researchers. And importantly, at this step, the medicinal chemists joined this project to improve antiviral activity and reduce cytotoxicity, and most importantly, to acquire patent right. So after evaluation of antiviral activity of the lead compound in an animal model, and also evaluation of pharmacokinetics and toxicity in animal model, we name this compound drug candidate as antiviral agent. This talk, I said before, I will introduce to you anti-influenza viral agent as a representative case of my antiviral research. This slide shows the influenza A virus life cycle. Virus enter the target cells through endosome-mediated endocytosis. After fusion, membrane fusion step, A-segmented viral genomes are released into cytoplasm and then migrate into the nucleus, where RNA-dependent RNA replication initiates. Currently, there are three classes of antivirals improved: NA inhibitors, including also tamivir, zanamivir, and IMT inhibitors such as amantadine and lumantadine, and recently approved polymerase inhibitors such as albigan, also named favipiravir, and also Xofluza. Even though there are different cases of antivirals highly effective against influenza A or B, still emerging of drug resistance mutants are still great concerns. So we need to develop another antivirus with alternative mode of action. This slide summarizes the current progress to develop antivirals commercial in pharma companies. It is about 5 years ago review, so it's too small. In this slide, T705 and this compound is Xofluza have been approved already, and also HA-specific monoclonal antibodies are also under development. Particularly, BEDI antibody are used as a control in this project. This slide shows the final result of my screening. I found after screening of about 7,000 compounds in a cell-based system, I found delidrin is active heat compound. It showed antiviral activity against influenza A virus consistently, but depending on strain-dependent manner against H3N2, but it has no antiviral activity against influenza B virus. So as a next step, as I said before, I collected delidrin derivatives from chemical bank. So I found they provide me about 7 alternate chemical derivatives. I also test antiviral activity again. The conclusion is that ifenprodel and clenbuterol showed a similar antiviral efficacy profile as delidrin. However, other compounds, rest 4 compounds had no antiviral efficacy. First, I investigated ligand-based pharmacophore modeling. These 3 compounds has highly potent antiviral efficacy. This first one is delidrin heat compound. The last 4 compound has no antiviral activity. This ligand-based pharmacophore modeling study clearly showed that among the core skeleton, these 2 positive electrostatic and this positive electrostatic lesion, as well as negative static region, could be highly important to maintain antiviral efficacy. Probably van der Waals domain maintained to make a hydrophobic interaction may be option. So our hypothesis has been proved by this negatively selected antiviral compound. As shown here, these 2 compounds has no positive static interaction, but they had no antiviral efficacy. Actually, after screening of antiviral compound, it is very dangerous. The heat compound is actually active because some compound, if some compound has color or fluorescence, they could affect antiviral assay system. So we need to first again confirm their antiviral activity in another method. First one is Western blot. First method is Western blot analysis. This result clearly shows that delidrin inhibits influenza A viral protein expression in cell level as efficiently as a control reference compound or oseltamivir or ribavirin. In a cell culture supernatant, viral titer also reduced in a dose-dependent manner. These 2 data clearly demonstrate that it has antiviral activity in infected cells. In time of addition experiment, we find that it could target viral entry step between 0 to 1 hour after infection. This confocal microscopy clearly visualized that this migration of vRNA into nuclear localization, has been blocked in the presence of delidrin. This result confirmed again this confocal analysis. We tried to co-localization lab 5 or lab 7 with viral MP protein. The last slide showed the case, the delidrin case. Most of viral protein, MP protein is red one. MP protein has been co-localized early endosome or late endosome markers. The results indicate that this delidrin block the viral entry step by arresting most of the vRNA particles in the early or late endosome particles. I will show you again the viral life cycle again. This is a viral entry step. This endosome-mediated the membrane fusion step. So I assumed at that time that delidrin could target this entry step as shown before, confocal microscopy. In this step, 2 viral proteins highly involved in this endosome-mediated endocytosis, including hemagglutinin and M2 proton channel. From this result, I could decide which protein is major target of delidrin. So I have to test this antiviral protein assay. In detailed description, this is endosome membrane and viral particle. In endosome, HA2 make a bridge between viral membrane and endosomal membrane. And in parallel, empty protein also as a proton channel, it facilitates proton influx from endosome to viral particle inside. These two proteins highly close and participate in viral endosome-mediated endosome membrane fusion step and also release of asegmented viral particles into cytoplasm. First, we test whether it could target empty protein or not. According to previously reported paper, we also established this proton channel activity assay system. This left slide showed that the black light show the empty channel activity. At acidic condition, proton channel is open, and also we can detect proton channel mediated fluorescent activity. And this channel, proton channel, is blocked in the presence of amantadine red one. However, the lead ray had no effect on proton channel activity, so we decide it does not target anti-proton activity. As a next step, we investigate whether it targets HA-mediated membrane fusion or not. This synthesis formation assay system clearly shows that at the acidic condition, in infected cells, synthesis formation has been efficiently detected. However, as I said, the lead ray HA2 specific monoclonal antibody blocked this synthesis formation, and also the lindrin has inhibited this synthesis formation in a dose-dependent manner and also as efficiently as the positive control HA2 monoclonal antibody. At that step, we determined the lindrin target HA fusion. And also in trypsin assay, this HA2 sensitivity was decreased in the presence of the lindrin in a dose-dependent manner. The data indicate that the lindrin blocks structural conformation change of HA2 and also subsequently block H2-mediated endosome fusion. This is the last one. This is the last one. Finally, we challenged it. We tested whether the lindrin is also active in an in vivo animal model. So we established the influenza A virus challenge model. Red one is show the virus infection mediated body weight decrease, and most of all mice were dead at day 11 after viral infection. However, co-administration of the lindrin completely blocked viral infection. 100% mice were survived after intranasal administration of the lindrin. This is my conclusion. The lindrin inhibits influenza virus. Its target is viral HA2 fusion protein. It also expressed its antiviral activity in an in vivo mouse model. I would like to thank my colleagues, and the antibody, antibody HA2 specific monoclonal antibody was provided by Dr. Sang-Soo Kim, correct? And this work was supported by CRIC and also Korean government. Thank you again.
Thank you, Dr. Kim. I really hope that your compound will develop as a new class of the antiviral against influenza. So I turn my microphone to Dr. Chekonin.
Thank you very much. Dear colleagues, let me introduce you Professor Sergei Netyosov. He is a corresponding member of Russian Academy of Sciences, as head of the Laboratory of Biotechnology and Virology, Faculty of Natural Sciences, Novosibirsk State University, Deputy Chairman, Special Counsel for Genetic Engineering of the Russian Academy of Sciences. With the presentation, coronavirus SARS-CoV-2, current situation, diagnosis, and vaccine development in Russia.
Good day to everybody. I was already introduced to you. My location is Novosibirsk. It is between Moscow and Seoul, and therefore I am in the middle of the huge distance between our countries. First of all, second, I would like to express my gratitude to organizers of this meeting for the invitation. And I also would like to say that I visited Korea 2 times in my life, and both times I was greatly impressed by this country, by the people, and I would be very glad to visit your country again. The title of my report is presented here. And first of all, I will present you the data about dynamics of daily cases of COVID-19 morbidity in Russia till 1st December. Unfortunately, we have a second wave of infection in our country, and this second wave is 2.5 times more intensive than the first wave, as you may see here. And unfortunately, we still do not know when it will start to diminish. I would like also to underline that the amount of diagnostic tests produced in our country is really very high, and every citizen of our country may easily give his sample for analysis. I may say it not only from internet publications, but I have a practical experience in this field because the most members of my family already had this infection. They successfully survived and are healthy now, but we had practical experience in this field. Now I would like to present my vision of current situation of diagnostics in Russia. And we have now 3 different systems of diagnostic. First of all, it is a PCR diagnostic in which we detect viral RNA. Second, we have test kits for evaluation of the presence of S protein or nucleoprotein proteins by ELISA tests. And second, we may test the infection by determining IgA or IgM antibodies after the 7th day of infection, and the past disease by the presence of IgG antibodies. Unfortunately, we have a huge amount of diagnostic tests registered in Russia. For the 1st September, we had 126 tests for antibodies to coronavirus, 31 tests for detecting viral RNA. And in addition to it, among these test systems, there were significant amount of test systems with express determination of antibodies or viral RNA. Unfortunately, some IgG antibody test system may give positive results if a person has recently been sick with one of the common 2 beta coronaviruses. Now, a few words about RT-PCR diagnostic targets in current coronavirus genome. The most of the RT-PCR test systems have the following 3 regions in the genome for the determination of RNA. It is, first of all, ORF1AB. Second, it is a E protein. Third, it is N protein, and S protein is not shown here, but some companies had chosen S protein gene 2. As you may see, the main part of detection sites are located in the right part of the genome. And usually it is okay. But when we start to determine the presence of viral RNA in the late stages of the disease, we may see that some patients, in some patients' samples, the positive signal may be obtained up to 19th 90 days after the symptoms. And it is still a puzzle whether the virus is still replicating in their bodies, or the virus is not replicating, but some fragments of the virus genome are replicating there. And it may be tested by the simultaneous test of samples from these patients from the right part of the genome and left part of the genome, because the presence of live virus is hard to show by experiments, and it is very expensive. A few words about diagnostics of antibodies. Antibodies may be detected to a few proteins of SARS-CoV-2 virus. Surface S protein, membrane surface E protein, and nucleoprotein. And the most abundant proteins of the virion are S protein and N protein. And it is on them that the largest amount of antibodies is produced in the host organisms. The easy way to develop the system is to develop the system with N protein, because it is not glycosylated, and therefore recombinant analog can be produced in large quantities in bacteria for application to a substrate in ELISA method. And purification of it is not difficult. Therefore, in the first stage, the overwhelming majority of ELISA test systems were based on N protein. And therefore, the antibodies from this protein were determined in such test systems. But the most abundant surface protein of the virus is the S protein, spike protein. And the virus neutralizing antibodies are produced against this protein. In any case, the antibodies against N protein are important too, as it had been shown in the research in a few papers. If the person has antibodies to N protein, it means that he has T-cell immunity. But the neutralizing immunity is engaged with S protein. Therefore, we should take into account every time when you use this test system or that test system, what is the antigen at this test system? What's the type of antibodies we evaluate by this test system? It is very important for this purpose to have control sera panels. Unfortunately, up to now, we have access for only one domestic control sera panel produced by the company Chema. And it is a good set of reagents, and it is being widely used now in Russia in the diagnostic companies to evaluate and to compare different test systems. And it is also very good control panel produced in Great Britain, which is mentioned here. But it is not widely used in Russia because it is hard to import it there. I hope very much that it will be available very soon in large quantities, and I'm sure that we would be very glad if some Korean control sera panels were available in Russia too. Here is the results. Here are the results of analysis of samples of corona control panel produced by Chema company in test systems of different manufacturers in Russia, and it is also one foreign test system. I intentionally do not name the companies, just numbers, because it is not an easy task to compare these test systems, because they have different calibration curves and they have different indicators to do it. But we may see, even from this preliminary, very preliminary picture, that test systems are quite different in quality. And we should know why this is such a difference, and how to select really reliable test systems For this purpose, we should have more sera panels, and we should have independent evaluation of these test systems, because in this case, we will know to what test system we should trust more. Now about vaccines. Here are listed main types of antiviral vaccines, and you, of course, know very well that we have live vaccines, inactivated vaccines, live recombinant vaccines, mRNA-based and DNA-based vaccines, peptide-based vaccines, subunit recombinant vaccines based on recombinant viral proteins, and viral protein-based vaccine. It means that producers use natural, non-recombinant viral proteins for such vaccines. It is a picture from a website, Coronavirus Vaccine Tracker, which is supported by New York Times. And you may see now that there are a lot of vaccine candidates in preclinical stage, 87. And a lot of vaccines are in different phases of clinical trials also. You may see the amount of them. And we already have 7 conditionally pre-approved vaccines. First of all, it is a Chinese recombinant vaccine from CanSino company. It is based on adenovirus serotype 5. This Russian vaccine, Sputnik 5, also recombinant, but based on adenoviruses 26 and 5 serotypes. And there are also 3 Chinese inactivated vaccines mentioned here. It is a S-protein peptide-based vaccine developed by State Research Center Vector, located, by the way, in the Novosibirsk, close to our university. And finally, Biotech Pfizer vaccine, which has been recently authorized in Great Britain. And there are additionally 12 vaccine candidates in phase 3 clinical trials. Among them, there are vaccines already mentioned in the previous slide, but I would like also to mention vaccine developed by Moderna company from United States, the vaccine developed by AstraZeneca together with Oxford University based on adenovirus, recombinant vaccine based on adenovirus type 26 from Johnson & Johnson. Again, 3 inactivated vaccines. It is very interesting trial being engaged in Australia with BCG vaccine. And I already mentioned our Russian vaccine Sputnik 5, based on 2 adenoviruses, and 3 other vaccines. 2 of them were mentioned, but recently Indian company Bharat Biotech designed an inactivated vaccine produced in India. Sputnik 5, it is a 2-component vaccine, as I already mentioned, and it is a brief description. And you may see that there is a very high concentration of virus there, mentioned here. The clinical trials phases results were published in the Lancet Journal on 4th September, and you may read it here. And in September, they were started post-registration clinical trials of this vaccine, and the results are mentioned here. And there are very positive results of this vaccine, and these results will be soon published in some international journals, and therefore we will see the exact data about it. But we have additionally a few vaccines in development stage, and very interesting one, which is not widely mentioned in the mass media. It is a live recombinant vaccine based on Russian Leningrad 16 measles vaccine strain expressing S protein. And the phase 1 clinical trials will start soon. And really, we have about 10 different vaccine technology being developed now in Russia. So we have very good perspective for the future. And first of all, I would like to mention that we have well-developed test systems against IgA, IgM, and IgG antibodies. But we need to compare all these test systems by specific sera panels. And with this purpose, we already are developing our own sera panels, but we'll be glad to receive these sera panels from another countries. As for vaccines, as I already mentioned, we have now a few candidates, and Sputnik 5 is in the most advanced stage because we already have preliminary results of phase 3 clinical trials, and they are very positive. Effectivity is about 95%. We have also some talks about testing some foreign vaccine in our country, which I mentioned here. And I would like to mention that we really need to have a choice for vaccine, because some vaccines will be good for young people, but some vaccines, more safe, will be good for elder people and people which are immunocompromised by in any way. So we have some very positive perspectives to struggle successfully with this infection. And in addition to it, we have developing and have developed already some vaccine technologies which will be very perspective in the future because they are quite fast, they are well tested, and these technologies may be used in the future. Thank you very much for your attention.
Thank you, Professor Joseph, for your brilliant presentation. And I would like to pass the word our Korean colleague.
Okay, I'm happy to introduce the last speaker of this session, Dr. Song Dae-sop. It is my understanding that Dr. Kovalenko, who was supposed to be the last speaker, was unfortunately not able to make the symposium. So this means Dr. Song will be the last speaker. And he is currently the professor of the College of Pharmacy at Korea University. He received DVM and his PhD of veterinary medicine in 2004 at the Seoul National University. And he's currently a member of board of directors at Korea Society for Zoonosis and the Korean Vaccine Society. He endeavored on working on commercializing an oral swine coronavirus vaccines, and he recently focused on the preclinical validation and development of several important viral vaccines. He will be speaking on nanobiotechnology for diagnosis, vaccine, and treatment against disease X. Dr. Song, please.
Yeah, it's a great honor for me to as a last speaker, and I also very happy to have a presentation in front of your attendance, because as I know, Russia is a very famous country in terms of virus history. As you know, Russia has Dmitry Ivanovsky, who found at the first time the TMV virus. So it's a great honor to have a presentation, especially on that virology topic. Actually, today, my main purpose of this talk is to share idea and to promote the joint research as an international collaboration. So rather than to introducing detailed result, I will show my recent performance regarding to the convergence study between nanotechnology and the biotechnology. As you see this slide, my expertise and research area covers the interspecies transmission, including corona and influenza virus, and to have a development, diagnostic, and the vaccine of the virus. This is a brief contents of this talk. And actually, as you know, recently, disease X is the very famous concept for understanding the infectious disease. As you know, the disease X means unexpected infectious disease. As you did these pictures, in 20th century, we faced repeatedly the pandemic, intervally 20 or 30 years. However, after the 21st century, every 2 or 3 years, we faced a very new emerging virus disease. Unfortunately, 1 year ago, the end of the 2019, now we are experienced another disease X. The first example of the disease X, the COVID-19, the very important message and the very important fact is until now, this is not, we cannot experience any peak. The peak is not the peak is not yet. So that's the main concern for the mankind. Actually, 2 years ago, WHO announced that disease X would be the great concern to the human race and mankind. As you see this slide, disease X as examples, disease X would be the Ebola and the Zika and MERS and another unexpected disease. Unfortunately, COVID-19 is the first real case of the disease X. The X means unexpected and emerging viral disease. Actually, there are so many sources of the disease X. However, the new emerging virus from the animal would be the real example of the disease X. Actually, interestingly, the 3 months ago of the real outbreak of the COVID 2009, WHO have an expectation that the first disease X would be the zoonotic and RNA, and the respiratory virus would be the first disease X. Unfortunately, the expectation is the real just right. So, because when we regarding this situation, we should be prepared the real tactical preparedness against the new emerging and another disease X. So, what is a fundamental reason of this kind of new emerging disease? So many people pointed that because of the rapid development of the wildlife, this result in the getting closer and closer between human and animal interface. So the main fundamental reason of this kind of new emerging disease is the human cross the line and human destroy the wildlife environment. And recently, the Nature published one article that showed that rapid urbanization increased the risk of the zoonotic transmission from the so many kind of species, including avian and rodent and some kind of pest species. And in addition to these reasons, also as you know, the industrialized livestock business, so big farming, the mass production system of the livestock industry result in the higher rate of infection, especially the virulence, hiring the virulence, and make many chance of the recombination and the immune suppression system. So, summarized with this kind of a reason and the fundamental reason of the human cross the line, this would be the fundamental reason of the emerging infectious disease. So, actually, the most important concept to understand the infectious diseases, especially the zoonotic disease, is the human-animal interface, the other words, speed of infection. That would be the future concerns as I previously described as a disease X. So this time, this is a cytopathic effect of the coronavirus infection. As you see this movie clip, within the 72 hours, this coronavirus destroyed the epithelial cell, and they induced a very typical cytopathic effect. And also, I want to emphasize the difficulty to prevent and dealing with the coronavirus. As you see this slide, coronavirus is very dynamic, and so many species have its own coronavirus infections, especially to the pigs and companion animals, and so many bats and the human also have so many coronavirus infection. In addition to this fact, the coronavirus have a very various target to replicate. For example, some coronavirus have a replication in the digestive system, and the others have a replication in the respiratory systems. That's why the dealing with the coronavirus infection is so difficult. As the last slide of the introduction part, as a summary of the clinical sign and affected area of the body. As I described the previous slide, summarized all the animals' characteristics. This COVID-19 virus, as we assumed from the title, or rampage through the body, they can attack the digestive system, they can attack the respiratory system mainly and the blood vessel mainly. However, they can have a many effect whole through the body. So that's why we have so many difficulty to prevent and cure these virus infections. And also, I want to emphasize one more concept of the reverse zoonosis. Do you have you heard about the terms reverse zoonosis? Actually, as the terminology, the reverse zoonosis is the infectious disease from the human to animal. The zoonotic is from animal to human. So we can find recently some cases of reverse zoonotic infections. So you should have pay attention to the reverse zoonotic cases. The examples of reverse zoonotic would be the ringworm or the mumps or especially the influenza viruses. Actually, according to our report recently, the influenza virus could be the reverse zoonotic infection, especially to the companion animals, as evidence of this slide. So that's why we should pay attention to the reverse zoonotic cases. Actually, although the COVID-19 virus is zoonotic viruses, however, in the relationship between the human and the companion animals, so many cases tells us that until now, these cases are all reverse zoonotic infection, not the zoonotic infection. So that's why we should pay attention to the reverse zoonotic cases of the COVID-19 cases. Yeah. Actually, I'm sorry, the very long introduction part. From now, I will briefly talk about my recent performance about the nano biotechnology. Actually, for the perfect prevention of the infectious disease and to make a preparedness, as you know, diagnosis and the antiviral and the prevention with the vaccine would be the best way to tackle with the new infectious disease. Actually, the nano biotechnology, nanomaterial is very easy to make a virus mimics in terms of size, in terms of structure. So that's why we have a choice to have a collaboration between the nano and biotechnology. I will show our recent performance about the diagnosis, especially for the pathotyping of the influenza virus. Actually, we use a very fundamental characteristic of the influenza viruses. Every influenza virus, when they attach to the cell and when they have power to have a replication, they should have cleaved with the enzyme in the body. Actually, however, the highly pathogenic viruses would be very easily cleaved with furin and trypsin enzymes, but the low pathogenic viruses have a limitation with the cleavage. And using this activity, we make traffic systems. We can easily differentiate between highly pathogenic and low pathogenic influenza viruses. Also, recently, when we have development of a COVID-19 vaccine candidate, using the self-assembly vaccine, we can promote the efficacy of the vaccine and immunogenicity vaccine using the polyethylene glycol and the polyl lactide complex. We can make a self-assembly vaccine, and now we are on the animal study for making the better vaccine compared to the traditional subunit vaccines. Also, in terms of adjuvant, we also make a cationic adjuvant, including squalene. Actually, at the first time, we have a mimics to the international adjuvant named NF59. However, we have some modification and got a patent, and also have evidence to have a very good efficacy after the real virulence challenge to the animal models. And also, in the swine models, we have oral vaccine adjuvant that can be have a great resistance against gastric acid. However, when they enter the small intestine, the adjuvant can promote the immunogenicity to the mucosal membrane. So now we are on the clinical trial in the pigs models. And also recently, although this is very preliminary, our trial for making the antiviral candidate, the same beaker as we used in the diagnostic system, inside this polymorphism, we inserted the commercialized antiviral. So we have a very advanced efficacy with the antiviral systems. And finally, recently we have fluorescence and the pigmentation to the virus directly. So we can have a simultaneous detection, and we can follow the movement of the virus movement. So at the first time, we apply this concept to the dengue virus. So as you see this slide, when we have a pigmentation to the virus directly with the modificeramide, that kind of fluorescence. So as you see this slide, we can simultaneously detection of the dengue virus movement. With this technology, we can screen the antiviral performance, and because this fluorescence can show the simultaneous movement of the virus, we can apply this technique to the screening the antiviral candidate. So combining of these previous all studies, diagnosis and prevention and treatment, recently we published one article combining of all these technologies. So if you have any interest about the prevention and the diagnosis and treatment for the new infectious virus, so just do not hesitate, and please let me know to have a joint research together. Thank you for your attention, and because of the time limit, I want to close my slide with this slide. Thank you so much.
Thank you, Dr. Song. Very comprehensive and intelligent use of nanobiotechnology for diagnosis and vaccine development. This pretty much wraps up our presentation, and probably we have about 20 minutes of Q&A and discussion. Dr. Chekhonin, on Russian side, would you lead the discussion?
Yes,
Well,
of course.
yes.
We will support your suggestion to start our discussion now with some points, very important points, and we can start the discussion. Are there any questions? Please, Professor Mukashev.
Dear colleagues, not really a question, but I would like to give a short comment, not about what has been said, but about what has not been said. We are now quite advanced in the outbreak, and we are researchers. We should be thinking in advance, and we should be now thinking about the exit strategies, about continuation strategies, because it is clear that one way or another, either by natural infection or by vaccination, most of people would encounter virus antigens soon. But frankly speaking, we do not have understanding what to do then and how the story would continue in 6 months or in a year. And one of the problems is that because we do not have reliable serological tests. Like Professor Nityosov noticed in his presentation, the tests that are currently on the market are not reliable. And I'm sure that he provided better tests, because our experience with so-called express tests for antibodies show that they can easily have a sensitivity below 50%. So there is a complete chaos on the market. There are no standards, and there is thus no understanding how many people encountered the virus. So panels are one thing, but I think that the golden standard of all testing is the neutralization test. So it is very important to couple all studies on antigenic tests with virus neutralization, because it is the measure of what these antibodies are. There are all kinds of cross-reactivities. So anything what is possible with antibody tests now. So I think that this is a technical part, which is one of possible fields of cooperation. I have witnessed that Korea is very efficient in assay development. For example, in our lab, we have been using the Korean PCR assay, which targets simultaneously 3 genes and has internal control based on human RNA. This is the best PCR test that we have seen and we have used, although it's unfortunately very expensive. So we have a lot of infected people. We have good clinical data, or at least relatively more abundant. And I see it as one of very important directions of future cooperation.
Thank you. You really have mentioned the importance of the diagnostic area, and I think if the diagnostic antigen is good, then the diagnostic antigen could be also applied for the vaccine antigen. So that means that that's a very good common platform to apply for 2 different directions, diagnostics and prophylaxis. Probably that is one area of the mutual collaboration between Korea and the Russian side. Do anybody wants to elaborate on vaccine area? Probably.
Professor Son, I would like to ask your personal opinion about future of different types of vaccines. What do you mean? What kind of type of vaccine has more perspective? What do you mean? What is your personal idea? Now we're discussing a lot of types of candidate vaccines, and now it is important to conclude what type of them has some advantage. What is your personal idea?
Yes, I'm not sure whether my answer would be satisfactory to you or not. And the current outbreak of COVID-19 is really all of a sudden. And the speed of spread globally is outpaced any speed of the vaccine development. Now the lesson we are having right now is to develop a common vaccine platform that would allow speedy delivery of the vaccine, the vaccine component. And probably one of the successful example of the COVID-19 era is why is the mRNA vaccine was so successful is that it could be produced within 2 weeks in the laboratory without any cell culture basis production system. The other one, vectored vaccine is quite successful, although there is a remaining question lingering around on the effectiveness of vectored vaccine, especially because of the pre-existing antibodies against adenovirus that is being used by AstraZeneca and other companies. But we really have experienced through this experience, we acknowledge that the fast delivery, the vaccine platform, is required to be prepared for any pandemics, including disease X, whatever it means, comes around to humans. And probably, if I focus the vaccine production platform with respect to the VLP vaccine platform that I presented today, is that the production of the recombinant antigen is much simpler and faster and easily scaled up through the bacterial system, as long as you do have a good folding and assembly system in bacteria. And I hope the bacteria system, I really think the bacterial system has been neglected for so far because of the lack of the folding and assembly system in the bacterial system. However, harnessed with the novel chaperone system, the VLP or nanoparticle and recombinant vaccine platform from bacteria may survive or even thrive in the race of the speedy delivery of pandemic vaccines. It remains to be seen. I'm not sure whether my answer is satisfactory or not, but this is presently my own opinion.
Thank you very much. Yeah, please.
Yes, dear colleagues, I would also give my opinion. So I think that we do not know about the quality of the current vaccines, and we are lucky that there are so many. I would personally prefer a vaccine that gives both T cell and antibody response, and I got already vaccinated with a Sputnik vaccine. So I have neutralizing antibodies, and I do not know, but I hope I have T cell clones. But I think that all vaccines that are currently in advanced clinical trials are not satisfactory in the respect that they do not provide mucosal response, or they're not guaranteed to provide mucosal response. Because a coronavirus replicates on the mucosa, and from the evolutionary point of view, or from what we know from respiratory coronaviruses, it does not really care about antibodies. So antibodies can prevent severe infection, hopefully, but most likely they would not prevent replication of virus on the mucosa. Which brings us back to live vaccines, to genetically engineered live vaccines. As far as I know, there are none in advanced clinical trials, but Professor Nityosov mentioned one on the measles virus platform. There are developments on influenza virus platform, and I think the second generation of coronavirus vaccines would include live vaccines, and they have much better future in a long perspective.
Yeah, I really agree with your comments that the mucosal immune response it constitutes a very important part of the protection, and as well as the CTL or T-cell based vaccine probably operating on the conserved antigen may provide protection against not only COVID-19, but also some maybe some genetic variants that would be able to arise in the near future. I wonder whether Dr. Song Jae-sop could comment on that? Yeah.
Yeah, thank you. Actually, as your comment, I really agree with your opinion about the importance of the mucosal immunity. And also, I want to say that actually, there in the virology history, there are so many cases of application of the coronavirus vaccine in the animal species, especially to the swine and the bovine and the avian species. So it's a very similar type of pathogenicity. So we should find some clue from the vaccination to the animal species, especially because it's a very similarity to the target. It would be the digestive or the respiratory mucosal system. So that's why we should have some experience and the evidence from the application of the animal coronavirus species. However, until now, because we found this virus within 1 year, there are so many we don't have many fact about the viruses. For example, we don't know the exact time of the exact duration of the immune response, and when we have a vaccination, we don't know the exact duration of the immunization. So that's why we have many trial and study about the immunity of the COVID-19 vaccine.
Actually, I have a brief question to Dr. Karpova. She actually shows very, very interesting result on the converting rod-shaped virus into spherical by simple thermal treatment. and as an RNA-free VLP type of vaccine component. My question is that you are mixed with the spherical TMV with the antigen of interest. I wonder how stable is that complex? Because I wonder whether the interaction between the spherical particle and the antigen may not be that strong. And I wonder how stable is that complex, and how wide, how many different antigens could be applied to your approach? Could you answer for that?
Yes, yes. Thank you very much for your question. Yes, we have shown that antigen interact with the surface of spherical particles by simple adsorption without any chemicals, without any bound agent. And we have some, okay, just a minute. We have, English word, one moment. We can present on the surface of spherical particles about five variants of proteins, recombinant proteins, 5 variants. It doesn't matter if their size of their, from what amino acid they consist of. And we demonstrated this effect with by immunofluorescence microscopy with different color fluorophores. Maybe we can present on the surface of spherical particles and more antigen than 5. But now we have data that 5, it is possible. Do you answer to your question?
Thank you very much. Yes, yes. Thank you very much. Yes.
Sorry, I have a question to Professor Nityosov. In your report, you were analyzing different types of vaccine and on different platforms. And what do you mean? What do you think? Has the so-called the Chumakov vaccine some advantage? You know, our registration system has already registered 2 types of vaccine on different platforms. And has that Chumakov vaccine some advantage or not?
First of all, I would like to mention that Chumakov Institute is the most experienced in development of vaccines among all the vaccine developers currently working on SARS-CoV-2 vaccine in Russia. In addition to it, they have a production unit. It is producing now vaccines against tick-borne encephalitis virus, against rabies, and yellow fever vaccine and polio vaccine, and they produce both live and inactivated vaccine. And they have very good methods of purification of viral particles. So I trust these specialists because of their real practical experience in this field, and I'm practically sure that inactivated vaccine will be one of the very good vaccine candidates. But of course, the phase 3 clinical trials results will determine its future in comparison with the results of other trials of other vaccines. So it has perspectives.
Thank you very much. Dr. Kaplan has.
Please, some words. I think that we need the different vaccines and on the different platforms now. We can develop the different vaccines. And only time will be judge what will be better. And with this connection, my special thanks to Professor Selong for their report, virus-like particles as pandemic vaccine platforms. It was very interesting for my lab. Thank you very much.
If I add one more comment on that, the front runners in the COVID vaccine is manifested by two different type of vaccines. One is, as you know, adenoviral vectors, and the other one is mRNA-based vaccines. But the traditional vaccine is a recombinant or protein vaccine, but which is rather slow in terms of development. And because virtually all the viral antigens are produced by animal cell or insect cell, that is costly and rather hard to scale up. Therefore, it takes time. If any a production system that would enable speedy delivery of antigens, then I think that production platform would be in par with the mRNA or vector vaccine in coping with the infectious disease. What about just switching into the antivirals? Because so far we have discussed on the vaccines. Can I ask a question to Dr. Mi-Hyun Kim? You have very nice data on identifying a novel class of the anti-influenza antiviral compounds, nilidrin. Could you elaborate a little more the type of the antiviral action, the mode of action? You briefly mentioned it works on the fusion, H2 type of fusion system. Could you elaborate a little more than that?
Yeah, actually, as you know, most of monoclonal based antiviral development is focused on HA2 because HA2 is a highly important viral entry step, and also it is highly conserved, so it could have broad spectrum as antiviral agent. As a small molecule-based antiviral stress system, this H2HA target or any target is highly reasonable because entry step it is also important, but in this case we can exclude the concern about drug delivery system because antiviral machinery occurs on the viral surface or cellular surface before viral entry step. So I think HA2 is one of the most promising antiviral targets. So that's one. Yeah.
Thank you. If I ask one more question on the antiviral side, question to Dr. Sung-Taek Kim. Yes, you have a very nice presentation on the nafamostat. Is it both anticoagulant and antiviral? Is that the compound or the different compound?
It's nafamostat is only used for, it's not camostat, but nafamostat can be used as an anticoagulant.
So anticoagulant and antiviral activity, is there some common mode of action that can be shared by these apparently two different type of biological activity? What's the common mode of action?
You mean the nafamostat and
Yes,
camostat?
yeah.
Both drugs inhibit the TMPRSS2 so that it can block the viral entry.
Oh, I see.
Yeah.
That's why it functions as an anticoagulant.
That's right.
It interferes with the coagulation pathway, and then it functions as antiviral. I thank you very much.
I have one question to Professor Kozlov. We paid a lot of attention to SARS-CoV-2 viruses and didn't discuss a new direction in virusology. It's a detection of retroviruses in human tumors. Professor, what is your opinion? Can we use some methods of RNA interference to block the activities of retroviruses in those tumors?
Currently, the work is going on using proteins of coding by endogenous retroviruses to treat tumors like memory gland tumors, etc. And of course, interfering RNase approach would be very useful in this respect either. But fundamentally, we should first figure out the role of endogenous retroviruses in human cancer, which is not completely resolved, I mean, at the present time. And we need more fundamental approaches like those which I discussed today, which includes the evolutionary virology approaches and evolutionary oncology approaches to figure out, how we fundamentally can tackle these problems. But RNA interfering could be one of the useful approach, I would agree.
If you can detect such type of viruses, of course you can develop two directions, targeting transport some biological activities to the tumor. I mean, first of all, one of them based on some antibodies to proteins, or the second one to use some remedies based on RNA interference. And both of them can be productive, I think.
Yeah, we also should study the role of this. It's known that some human genes originated from endogenous retroviruses, and they're functional in human placenta, etc. And so it's like, again, it's like we discussed at this conference, I think Professor Lukashov discussed this gene clouds in respect to evolution of viruses and new recombinant form of viruses, and evolution of humans and other multicellular organisms have also kind of gene clouds, although the time periods during which these gene clouds interact with human bodies, they of course millions of years. Although the endogenous viruses like HML2, they infected human species. They may still be infecting, so that's why we should study them.
Can I ask one more question to Dr. Karpova one more time? Yeah. Yes. Your data on the spherical particle shows that you use the spherical particle to display the antigen of interest on the surface of the particle. Have you ever considered another possibility that you can encapsulate the foreign antigen into the spherical particle if you mix together somehow and control the reaction in such a way that encapsulate antigen of interest in the spherical particle? That would also work as a vaccine strategy.
Thank you for your question. If I understand it, when we prepare our complex consists of spherical particles and antigen of pathogen, we mix this in water at room temperature about 20 minutes. We have controlled this process by different approach, by immunofluorescent microscopy with antiserum, specific antiserum to pathogen, and second antibody with fluorophores, or we can introduce fluorescence marked in antigen and control this process by fluorescence spectrophotometry. Yes, I answered to your question. No?
Thank you for comment. Thank you very much.
Thank you.
I think we should complete our discussion. I would like to thank all the participants with very informative presentations. I would like to thank organizers of our meeting. I think our meeting will be very useful for both sides, and we will start some types of cooperation between different institutes in some important directions. Thank you very much.
Yes, thank you very much. And also from Korean side, it is my real privilege to co-chair this important session of viruses and epidemiology and immunology with Dr. Chekhonin, whom I respect most. I would like to thank for all the speakers from Russia and Korea and sharing their recent discovery on infectious disease, including COVID-19. I myself have long collaboration with Russian scientists, especially in the Institute of Experimental Medicine, St. Petersburg, on developing influenza viruses, influenza vaccines. And Dr. Rudenko, Dr. Galina Alexandrova, they are so eminent scientists who developed the world's first live alternative influenza vaccines. It has been developed much earlier than in the United States and commercialized in Russia. And I hope the Science Day between the two countries would serve as an opportunity to further our collaboration to better control COVID-19 as well as other viruses threatening us globally. And as cold winter approaches, some experts warn on the potential twin-demic, influenza and corona. And we probably, our scientists in both Russia and Korea, will unite together and work together to develop universal vaccines against a twin-demic situation. And hopefully, by the time we meet together next time, we may be able to talk each other face to face without any facial masks. And thank you, Dr. Chekhonin and my Russian colleagues. Thank you very much.
Thank you very much, Professor Son Violin. I think we had a really very interesting discussion, and I think we have a lot of possibilities to continue our work on a high level without the masks, and I think we will start our cooperation in a different level, and I hope we will do our best to develop our relationship. Thank you very much.
Thank you very much. Bye-bye.